In today's video, we'll be studying the cell nucleus, and here we have the outline of all we'll be discussing: the nucleus, the nuclear envelope, the nucleoli, the nucleoplasm, the cell cycle, DNA replication, mitosis, protein synthesis, cell renewal, telomeres and telomerases, apoptosis, karyotype, and clinical correlates. So we have a lot to discuss in this video. So starting off with our first outline of the video, which is the cell nucleus. So earlier on we discussed the cell plasma membrane, as well as the cytoplasm. So in this video we'll be discussing all about the nucleus. Now the nucleus is the central structure of the cell. Okay, and we can imagine it as the control center of the cell. It's kind of like the CPU of the computer, the central processing unit, and indeed it's a central structure. Okay, it's more or less the largest organelle that we have within the cell. Now, when we speak about the nucleus, there are three major nuclear elements. Now, the nuclear elements are: firstly, we have the nuclear envelope. Next, we have the nucleoplasm. And lastly, we have the nucleoli or the nucleolus. If it's one, it's the nucleolus. If we have more than one, then it's nucleoli. The nucleoli or the nucleolus literally means little nucleus. Okay, so this means little nucleus. We're considering each of these nuclear elements individually. For now, it's important for us to take note of the fact that the nucleus is that central organelle, dense. It's kind of like the central processing unit. It contains more or less all of the DNA of the cell. Of course, we discussed the mitochondria and we said the mitochondria also has its DNA. Most of the DNA of the cell dwells within the nucleus. The nucleus is kind of like a genetic library. All the information needed in terms of protein synthesis and so on dwells within the nucleus. So firstly, let's consider the first nuclear element, which is the nuclear envelope. Being the genetic library is a very sensitive area. Generally, when we have an organization, whatsoever function the organization may be carrying out, there's usually what we call integrity of information, and there's a bit of secrecy to the information possessed by such an organization. And it's also very similar to what we have in the cell. Okay, so the nucleus is this information center, and being an information center, it has to be bound by a membrane. And so the membrane that bounds the nucleus, it's called the nuclear envelope. And it's called a nuclear envelope because it's a double membrane. Okay, kind of like we have double security just to protect this information that dwells in the nucleus. Okay, so it's a double membrane envelope, and similar to the plasma membrane, it also has pores. Okay, so we have these nuclear pores. Now these nuclear pores, as we would go on to see, allow for instance, the RNA to leave the nucleus. It allows for proteins to enter into the nucleus. When we were discussing the ribosomes in our very first video on cells, we discussed that ribosomes are more or less macromolecules. Okay, so they have rRNA, ribosomal RNA, that is complexed with protein. Now the protein that forms the part of the ribosome actually is imported into the nucleus, then it combines with the ribosomal RNA, and then it's exported in form of subunits to become the ribosomes. Okay, so we'll see all of this in details as we go on. However, it's good for us to take note of the fact that firstly, the nuclear envelope is a double membrane structure, and similar to the plasma membrane, it has pores that allows for importation as well as exportation. The next that we have would be the nucleoplasm. That is the next nuclear element, the nucleoplasm. Similar to the cytosol, it's also a semi-fluid, translucent or semi-transparent. It has both colloidal and true solution properties. Within the nucleoplasm, we have some essential ions dissolved there. We have some essential salts, sugars, some proteins are also dissolved within the nucleoplasm. Now, very importantly for us to take note of is the fact that the chromatins, okay, so we'll go on to see the chromatins as we progress in our study. So the chromatins are also within the nucleoplasm, just the way we have organelles within the cytosol or the cytoplasm. We also have these chromatins within the nucleoplasm, and the chromatins could be in the extended form. So we could have extended chromatins. When we have extended chromatins, they are called euchromatins, okay, euchromatins. And when they are condensed, so they could be condensed chromatins, and when they are condensed, they are heterochromatins. Now, as we'd go on to see, when a cell is in the interphase, okay, so when a cell is not dividing, it's simply carrying out normal metabolic activities, we'll get to see that some of the chromatins are extended and some are condensed. Now, the partial extension and partial condensation of this chromatin is what gives the nucleus its chromatin granules. So we generally hear the term chromatin granules, and this granular appearance of the chromatin is due to the partial extension and partial condensation of the chromatins. Also, very important for us to take note of is the fact that the extended chromatins are those chromatins that are being actively transcribed, while the condensed chromatins are not being actively transcribed. Okay, so we would of course get to see all of this in details as we go on. Now let's take a look at the nucleus. Okay, so we have the nucleus, which we earlier stated simply means little nucleus. Now this is very interesting, and it's important for us to take note of this, that the nucleus is not membrane bound. However, the nucleus of course is bound by the nuclear envelope. Okay, now the nucleus is also dense and it's within the nucleus, and the nucleus is actually the structure that synthesizes ribosomal RNA. Okay, now just before we proceed, it's important for us to take note of the full meaning of these two terms. So we have RNA and DNA. Now RNA and DNA are polymers of nucleotides. Okay, they are polymers of nucleotide. RNA is ribonucleic acid, and DNA is deoxyribonucleic acid. As I stated earlier, they are polymers of nucleotides. Okay, polymers of nucleotides. Now, nucleotides generally contain a sugar, a 5-carbon sugar, and then in addition to the 5-carbon sugar, they have a phosphate backbone, as well as the nitrogenous bases. Now, the 5-carbon sugar could be ribose. Okay, so when it's ribose, we have RNA, or it could be deoxyribose. Now, when it's deoxyribose, we have DNA. So one major difference between DNA and RNA would be that while DNA contains the 5-carbon sugar deoxyribose, RNA contains the 5-carbon sugar ribose. We stated earlier here that a nucleotide contains the 5-carbon sugar, the phosphate backbone, as well as the nitrogenous base. So of the nitrogenous bases, we have two major divisions under the nitrogenous bases. So we have the purines and the pyrimidines. Okay, now for the purines, we have adenine and guanine. For the pyrimidines, we have cytosine, we have thymine, and then we have uracil. Now, adenine would always pair up with thymine, okay, and guanine would always pair up with cytosine. However, this is in DNA. In an RNA molecule, A, which is adenine, would pair up with uracil, U, and G would of course pair up with C. So this is another difference. The first difference is in DNA we have deoxyribose, in RNA we have ribose. Okay, in DNA we have adenine pairing up with thymine, and then in RNA we have adenine pairing up with uracil. Okay, so these are the nitrogenous bases that we have and of course we have the phosphate backbone. So it's important for us to take note of those two terms because we'd see a lot of that as we go on in our study. Alright, so far we have considered the nucleus, we have considered the nuclear envelope, which we said is a double membrane structure that contains nuclear pores or that has nuclear pores. We also considered the nucleoli or the nucleus as well as the nucleoplasm. Now very quickly before we consider our next subtopic, which is the cell circle, it's important for us to take note of a very important structure. Now this very important structure is the nuclear lamina. The nuclear lamina is a fibrillar structure and it forms a network of proteins. And the proteins that form the nuclear lamina are called lamine. Okay, lamine protein. Now if we have this to be the nuclear envelope, okay, so we have the structure in white to be the nuclear envelope. The nuclear lamina would be this fibrillar structure that forms on the interior surface of the nuclear envelope. So we would have it kind of like this. Okay, this would be the nuclear lamina. It forms a fibrillar network of proteins on the interior surface of the nuclear envelope. Now why is it important for us to take note of this? It's important for us to take note of this because the nuclear lamina not only provides support for the nuclear envelope, it also provides a form of attachment for the peripheral chromatins. Within the nucleus, we have some chromatins that just are on the peripheral surface of the nuclear envelope, and the nuclear lamina actually helps to provide kind of like an attachment for peripheral chromatins. Also very important for us to take note of as we would go on in our study is that a mutation could actually occur to the proteins that form the nuclear lamina. Okay, so we said the protein that forms the nuclear lamina is referred to as the lamine. There's a rare disease condition called progeria. Now progeria is a rare disease condition that is associated with abnormal cell aging and it could result in death. Of course, we won't be focusing on this disease condition in this video. However, it's very important for us to take note of the fact that the nuclear lamina is a very important structure, and a mutation to the protein or a defective protein in the nuclear lamina could actually result in abnormal cell aging. It's associated with slow growth, thinning of the hair, brittle bones, and these conditions generally result as one age. Okay, as people age, as people advance in age, especially old age, some of these conditions appear. However, the reason why it's an abnormal cell aging is because it actually occurs in little children, okay, and it's also associated with severe cardiovascular conditions as well as arthritis. It's important for us to take note of that. Now, of course, our next subtopic for this video, going by our outline, is the cell circle. So let's take a look at the cell circle very quickly. Now, the cell circle simply refers to the various activities that go on within a cell from the time it is formed to the time it reproduces. Okay, so from the period of formation to reproduction, all of the activities that go on within the cell, okay, within this period. Now, the cell circle is divided into two broad phases, okay, so the cell circle is divided into the interphase and the cell division. The cell division is associated with cell reproduction. However, the interphase is associated with the normal life activities of the cell, so the normal metabolic activities of the cell. The interphase is usually referred to as the resting phase of the cell. However, in actuality, the cell doesn't rest. Literally, the cell doesn't rest in the resting phase. The cell actually undergoes a lot of metabolic activities like protein synthesis and a lot more during the interphase. And so a more appropriate and a more accurate term for the interphase is the growing phase. So the interphase is the growing phase of the cell, or it could also be referred to as the metabolic phase. okay, because there are a lot of metabolic activities going on within this phase. Now, the interphase has some subphases. Okay, there are some subphases of the interphase, which of course we'll consider in this video. Firstly, we have the G1 phase, then we have the S phase, and lastly we have the G2 phase. Now, G means gap, so we have gap 1. S stands for synthesis, and G2 stands for gap 2. Okay, so we have gap 1, synthesis, and then gap 2. And as I stated earlier, the interphase is simply associated with the normal metabolic activities of the cell in the absence of reproduction. Okay, so in the absence of cell division. All right, so we have the gap phase as the interval between mitosis and synthesis. It's the period from when the cell is formed to the period just before it's about to reproduce. Before reproduction is DNA replication. DNA replication takes place around the period just before a cell begins mitosis. Okay, so just before a cell begins its mitotic division, DNA replication takes place, and DNA replication takes place at synthesis phase of interphase. Okay, so it's from formation, which is associated with mitosis, to DNA replication, which is just before reproduction. Now, of course, as I stated earlier, the synthesis phase is associated with DNA replication. Now, of course, we'll take a look at DNA replication in this video, but it's important for us to take note of the fact that DNA replication takes place at synthesis, or at the synthesis phase. Now, why exactly is DNA replication important? Now, as we stated earlier, the nucleus is that information center of the cell, so it contains all the information that the cell needs in terms of protein synthesis and so on. Now, it's important for the cell to be able to give its daughter cells this information, and the only way in which the cell can be able to transfer this information to the daughter cells is by replication, and so DNA replication is very important. Of course, we have chromosomes. Okay, we'll get to see chromosomes as we go on to study mitosis. Now, when we were considering the nucleoplasm, we saw, or we considered, the term chromatins. We have extended chromatins and we also have condensed chromatins. Now, the chromatin contains about 30% DNA. It has about 60% histone proteins and about 10% RNA. During mitosis, you have the chromatins condensing into chromosomes. However, when the cell is within the interphase, just carrying out its normal life activities, of course, we have chromatins. Now, the basic unit, or the fundamental unit of the chromatin, is what we call the nucleosome. Okay, now the nucleosome is made of histone proteins, so histone proteins and DNA. Very important. Now, the DNA in and of itself is a very large and long molecule. We need this DNA to be able to fit into our microscopic cell. Now, this is where histones come to play. Now, histones help to kind of like pack the DNA. So the DNA curls around the histone protein. So we can imagine the histone proteins to be something like this. And then we have the DNA coiling round and around the histone proteins. Something like this. This drawing doesn't do justice to the idea, but of course, it curls around the histone proteins in a manner that looks somewhat like this. And when we have the DNA and the histone proteins in this manner, what we have is the nucleosome, an octamer. 8 histone proteins come together to form the nucleosome. We mentioned the idea of DNA replication, and we said DNA replication is associated with the synthesis phase of interphase. Now, after the DNA has been replicated, so we have 2 DNA strands, and of course we know that the DNA has this double helix structure. The double helix structure, when the DNA replicates by a semi-conservative conservative method. Now this simply means that each of the DNA molecules, each of the two DNA molecules that would be formed would have one old strand, so would have an old strand and one new strand. So if this is the initial DNA molecule that we have, would have this old strand, and then the new strand would be, say, in yellow, so we would have the new strand looking something like this, and then the old strand would also be there present. Okay, so it has this double helix structure. Now when these two DNA molecules are formed, of course we would need the histone proteins. Okay, so we would need the histone proteins so that the DNA molecules can be wrapped around the histone proteins. Now the histone proteins are proteins, of course, and they have to, proteins are synthesized within the cytosol. Okay, so it means that these proteins would have to be imported from the cytosol into the nucleus, because DNA replication takes place at interphase, and at interphase the nucleus is still intact. Okay, so the nucleus is still intact, so we would need to have the histone proteins being imported into the nucleus so that the DNA molecules can be wound around them. Okay, so it's very important for us to take note of that. And then of course we have the gap 2 phase. So the gap 2 phase is the interval from synthesis to mitosis. Remember, we considered the gap 1 phase as the interval from mitosis to synthesis. Now the gap 2 phase is the interval from synthesis to mitosis. So what we have is that we have the DNA replication, which is associated with synthesis. So it's from the point where DNA begins its replication to the point just before reproduction, okay, which is associated with cell division. When we're considering reproduction in this sense, reproduction in the sense in which we're considering is cell division. So from DNA replication to cell division, this is the gap 2 phase. Okay, it's very important for us to take note of this. Now coming back to the cell cycle, which we said is divided into two main divisions. So we said the cell cycle is divided into the interphase, where we said the cell is simply carrying out its normal metabolic activities, and then we said the next step is the cell division step. So let's take a look at the cell division step of the cell cycle. Okay, so here we have the cell cycle, which is divided into the interphase as well as the cell division. Then we have the interphase divided into gap 1, synthesis, and gap 2. Now the cell division period of the cell cycle is associated with mitosis. Okay, so it's further divided into mitosis and cytokinesis. Now a lot of times cytokinesis is simply grouped together with mitosis. However, cytokinesis is actually a different step from mitosis. Mitosis has the prophase, metaphase, we have anaphase, and then we have telophase. Now cytokinesis actually starts from late anaphase and it goes beyond telophase, so it goes beyond mitosis. So we'll take a look at each of these phases individually. Alright, so far we have briefly considered the cell cycle. Now our next subtopic would be DNA replication before we consider mitosis, and then we go forward in our study. So let's briefly consider DNA replication. Now for this video we'll be considering DNA replication under 4 main headings. Okay, so the first heading would be the uncoiling. The next heading would be separation. Then we have the third heading to be the assembly, and lastly we have restoration. Okay, these are the 4 headings of DNA replication that I would be considering for this video. Now let's take a look at what goes on during the uncoiling phase. So as earlier stated, we said that we have the chromatins. Okay, so we have these chromatins, and we stated that the fundamental unit of the chromatin is the nucleosome. Also, we stated that the nucleosome is made of these histone proteins and DNA, and we said that the DNA is wound around the histone protein. Now just for us to be able to visualize this is how the histone proteins act. Okay, so the histone proteins act as a spool. They are spool like proteins. Okay, here in this picture we can see this is a spool, and wound around the spool is cable. Okay, so we have this cable wound around the spool. Now this cable could be several meters long. However, we can see that because of the spool, the cable is able to be wound around and it's able to be compact, occupying minimal space. This is how the histone proteins function with regards to the DNA. Okay, so back to our study. Now we have this histone proteins as well as the DNA. Okay, so what happens in the uncoiling phase? Of course, we're taking a look at DNA replication. All of these processes take place in DNA replication. So what exactly happens during the uncoiling phase? Now, during the uncoiling phase, we have the DNA molecule unwinding. Just to visualize it, we would have this cable unwinding from the spool. When we have this cable unwinding from the spool, that is basically how the uncoiling takes place during DNA replication. Okay, so now we have the DNA that is unwound. Okay, of course, this unwinding of the DNA is catalyzed by enzymes. Okay, so we have enzymes carrying out, facilitating this unwinding process, and during this uncoiling phase, we have the formation of a replication bubble. Now, the next phase is the separation phase. So now our DNA is unwound. Then we have the separation phase. Alright, so earlier we established that the DNA has a double helix structure and it looks something like this. Okay, this is the double helix structure of the DNA. Now, what exactly takes place during the separation? Alright, so we have our DNA, double helix, something like this. What happens during separation is that we have these two strands literally separating, and of course, this is also catalyzed by an enzyme. We're not exactly focused on all of the enzyme that are involved in DNA replication. However, it's good for us to take note of the fact that the enzyme helicase, okay, helicase is actually involved in the separation process. Firstly, we have the unwinding of the DNA from the histone proteins. Then we have separation of the individual DNA strands. So we have something that looks like this. Okay, so this would be the double helix, and we can see that at this point it begins to separate. Okay, so we can see the separation as it goes on. Now, during the separation, what we have is the formation of the replication fork. Okay, so we have the replication fork at this place. Of course, we have our nitrogenous bases separating. Okay, so we have separation taking place in this manner. Now, the next step in the DNA replication process is assembly. Okay, assembly is the next step, and the enzyme involved in this step is the DNA polymerase. Now, earlier when we started the video, we stated that DNA and RNA are polymers of nucleotides. Okay, we stated that DNA and RNA are polymers of nucleotides, and we said that these nucleotides of course have the sugar and phosphate backbone, and then of course we have the nitrogenous bases. So it makes sense that since DNA is a polymer of nucleotide, when we have an assembly of DNA, a polymerase should catalyze such a reaction. So the DNA polymerase actually catalyzes this reaction, and how it works is that it brings together free nucleotides. So we could have a free nucleotide with the sugar phosphate backbone and then the nitrogenous base. So this could be adenine, for example, adenine. So the DNA polymerase, it brings this free nucleotides to the separating strands. Okay, of course, we also have it on the other side. So we have something like this. This is catalyzed by the DNA polymerase. We have a single DNA polymerase for this side. So this would be the DNA polymerase. Okay, so we have our nitrogenous bases all through. Okay, we have our nitrogenous bases like this. So what we have is the DNA polymerase. So indicated here in red, the DNA polymerase helps to bring the free nucleotides. So this would be a free nucleotide. Alright, so we have the addition of these nucleotide bases all along the separated strand. Okay, so we have another nucleotide base and another nucleotide base. Of these two strands, we have the lagging strand. Okay, so we have the lagging strand and we have the leading strand. Now, we won't be going into the details of what a lagging strand is and what a leading strand is, but it's just important for us to take note of these two terms. So, of course, on this side, we also have the DNA polymerase attaching the free nucleotides in this manner. Now, of course, this could be adenine to thymine and then guanine to cytosine. Now, it's very important for us to take note of this. We have what we call complementary base pairing and we have obligatory base pairing. Now, complementary base pairing, adenine is a complementary base to thymine, guanine is a complementary base to cytosine in a DNA molecule. So complementary base pairing has to do with the fact that adenine would pair with its complement, which is thymine, and guanine would pair with its complement, which is cytosine. Now, what exactly is obligatory base pairing? Obligatory base pairing is very similar to complementary base pairing, but when something is obligatory, it's a must, it's an obligation, it must be done. Now, obligatory base pairing has to do with the fact that adenine must always pair with thymine and guanine must always pair with cytosine. It's an obligatory base pairing. Now, of course, when we were taking a look at the assembly here, we saw that we simply have the DNA polymerase attaching all of these free nucleotides to the old strand. Okay, so of course we have our old strand in green, so this would be the old strand, and then we have our new strand forming here in red. Okay, so we have the new strand. Now, it's very important for us to take note of the fact that because of the obligatory and complementary base pairing that we have, if we have an adenine on the old strand, the DNA polymerase would obligatorily attach to adenine thymine, okay? And if we have, say for instance, cytosine on this side, the DNA polymerase would obligatorily attach a guanine to it, okay? So this is the idea of obligatory base pairing. So of course, we have considered so far uncoiling, separation, as well as assembly. Now, taking a look at restoration, what exactly is restoration? So we have all of these free nucleotide bases attaching to the old strand. Okay, now it's important for us to take note of the fact that we have something like this basically. So we have this to be the old strand, and say for instance this to be the new strand. Of course, they are forming from the DNA polymerase. The DNA polymerase is attaching all of these new nucleotides. So we have our nucleotide bases from the old strand. So this would be old and this would be new. So we have all our nucleotide bases or a nitrogenous bases in this manner, and the newly formed nitrogenous bases, something like this, attached in this manner. And we said there's obligatory base pairing. Now, between adenine and thymine, the bond between these two nitrogenous bases is a hydrogen bond, as well as between guanine and cytosine. And of course, when we get to the RNA molecule where we have uracil replacing thymine, also between adenine and uracil, we also have the hydrogen bond. Okay, so we have the hydrogen bond in this manner. It's important for us to take note of the fact that we basically just have these nucleotide bases attaching. Okay, and we may have some gaps. Okay, so we may have gaps like this and gaps like this and gaps like this, representing this in a better way. So this would be the old strand. Okay, just to drive the point home, this would be the old strand with all of its bases. Now we would have, of course, the DNA polymerase just attaching the complementary bases in this manner. Okay, so this would be a free nucleotide, another free nucleotide. So we could have all of these gaps. Now, what takes place in the restoration phase is that we have an enzyme called ligase. Now, a good way to remember this enzyme ligase is to remember the word ligate. So ligate means to tie up. So from ligate we have ligament and so on. Ligament also kind of like ties up bones that form a joint. So ligase is, or ligase as you may pronounce it, is an enzyme that kind of binds these free nucleotides together. So it bridges any of these gaps that may be present. Okay, this is what happens in the restoration phase. And of course, it restores the double helix structure. Now, earlier we stated that, of course, we have these histone proteins. The histone proteins would then be imported from the cytosol into the nucleus so that the newly formed DNA can be wound around it. A very important term we also mentioned earlier is the idea of the semi-conservative replication of DNA. Okay, so very quickly, just to go over the individual steps or stages that we have in the DNA replication. First, we considered uncoiling, where we said that the DNA uncoils. Okay, so initially the DNA is wound around the histone protein, it uncoils. Then the strands that were initially bound by hydrogen bond, we had the bases bound, they separate. So we no longer have that well-defined double helix structure. Then the enzyme associated in the separation would be the helicase. Then we went on to the assembly, where we have the DNA polymerase that attaches these free nucleotides to the old strand. And of course, we have the restoration, where we considered ligase or ligase as you may pronounce it, which helps to splice together short DNA fragments. Okay, so this is a DNA replication, and of course now we have two newly formed DNA molecule. Okay, so we have considered DNA replication. Now let's take a look at mitosis. We had already started a study on mitosis, and we said we have the prophase, we have the metaphase, we have the anaphase, and we have the telophase. Okay, now we'll consider each of these processes individually, but not in details. Okay, so what takes place during prophase? Now, earlier we considered interphase, okay, and we saw the G1, we saw the S, and we saw the G2. It's important for us to take note of the fact that centriole replication is completed in the G2 phase. Okay, now the centrioles are the organelles, the structures that form the spindle fiber, which is a microtubule. So in the prophase, we have these chromatins, which we initially had as chromatin granules. We have them condensing. Okay, so they condense into chromosomes. So we have from chromatin granules to chromosomes. Now we have these chromosomes being made of two sister chromatids. Now as the chromatin condense, it's important for us to take note of the difference between chromatin and chromatids. Okay, as the chromatin condenses and forms this chromosome, what we have disappearance of the nuclear envelope. So the nuclear envelope begins to disintegrate and disappears. Okay, we also have the disappearance of the nucleoli. Okay, so we have condensation of chromatins into chromosomes, which are made up of two sister chromatids. We have the disintegration of the nuclear envelope, as well as the disappearance of the nucleoli, okay, during the prophase. Also very important for us to take note of is the fact that the centrioles begin to migrate at this point. So the centrioles begin to migrate, and as they migrate, they form spindle fibers. Okay, spindle fibers that we have here. So basically we have something like this. So initially we have our cell this way, and then of course we have the nucleus. Very close to the nucleus we have these centrioles at right angle to each other, and then they replicate. Okay, as they replicate, they begin to move to opposite poles. So we would have something like this. Alongside the replication of the centrioles, which actually takes place in the gap 2 of interphase, we have this disintegration of the nuclear envelope as well as the nucleolus. So initially we had the nucleolus like this. Okay, we also have the separation of the centrioles. And of course, we have a condensation of the chromatin. So now we no longer have the chromatin granules all around. What we now have is chromosomes. Okay, so we can see the chromosomes made of two sister chromatids, something that looks like this. Okay, so this takes place in prophase. Of course, later on in prophase, we have all of the formation of the spindle fiber. The spindle fiber begins to interact with the chromosomes that are being formed, and then of course we have metaphase. Now at metaphase, what we have is that we have these chromosomes lining up at the equator of the cell. Okay, so we have the chromosomes, something like this, and of course we have our centrioles. Our centrioles have now arrived, one each pair at opposite poles of the cell. We have something like this, and then we have the spindle fiber. Okay, and of course we have our chromosomes arranged at the equator of the cell, forming the metaphase plate. Some of these spindle fibers interact with the chromosomes. Okay, the spindle fibers that interact with the chromosomes are referred to as kinetochore microtubules, and those that do not interact are the non-kinetochore microtubules. And this is simply because the point of attachment of the spindle fiber, which is made of microtubules, to the chromosome is at a site called the kinetochore. Okay, so we have this arrangement at the metaphase. Now at anaphase, what takes place is that we have a shortening of the spindle fiber. So as the spindle fiber shortens, they pull apart the two sister chromatids. Okay, something that looks like this, and they continue to shorten. As they shorten, they take away the two sister chromatids, moving them to opposite poles of the cell. Of course, initially we had our chromosomes made of two sister chromatids. Okay, so these were the chromatids, and they were joined together at the centromer. Okay, so what we have is a pulling away of these two sister chromatids in this manner. The late anaphase, what we have, of course, we have a continual shortening of this spindle fiber, and as I stated earlier, cytokinesis actually begins at the late anaphase. Okay, and of course we also have cytokinesis associated with cleavage. Okay, so what we would have a constriction of the cytoplasm, something like this. So we would have a constriction of the cytoplasm ahead of the division of the cell. Now at telophase, what we have is that we have these chromatids. They have already arrived at opposite poles, and we have further constriction. Okay, something like this. It's also associated with the appearance of the nucleoli. So we have our chromatid, they have already arrived at the opposite poles. The spindle fiber become disintegrated. Then we have an appearance of the nucleoli. It begins to form gradually. We have the nucleoli forming here gradually. Of course, we have our centrioles, which have been replicated, and we have further constriction to form the cleavage furrow. And we said a particular filament actually acts during this cytokinesis that overlaps into and even beyond telophase, and that would be the actin filament or the microfilament. Okay, so of course we'd have further constriction until we have just two daughter cells. Of course, this video is not detailed, it's just kind of like a general overview of what exactly happens. So basically we have prophase, we have metaphase, we have anaphase, telophase, and then of course cytokinesis. Alright, so far we have considered the nucleus, the nuclear envelope, the nucleoli, the nucleoplasm, the cell cycle, DNA replication, as well as mitosis. Now let's take a look at protein synthesis. Earlier we considered the nucleus, and we said of course we have chromatin granules. Now these chromatin granules contain DNA, They contain about 30% DNA, about 60% histone proteins, and then about 10% RNA. Now, DNA, as we stated earlier, is a polymer of nucleotides. Okay, it's a polymer of nucleotides. Now, of course, we already established that the nucleotide is made up of the sugar and the phosphate backbone, as well as the nitrogenous bases, where we considered adenine, guanine, thymine, and then cytosine. And then we said thymine is replaced by uracil in the RNA molecule. Now, it's very important for us to take note of an important term, which would be genes. Now, what exactly is a gene? This is somewhat a standard definition of a gene, but it's important for us to take note of it. Now, a gene is defined as a series of nucleotide base sequence that encode a functional RNA. Simply put, a gene is a part of a DNA molecule that is used to form the template for an RNA molecule. And as we would go on in our study, we would understand what a gene is better. Okay, so basically we have the DNA. Okay, and we said this DNA is a double helix structure, something like this. And we have all of these nucleotide bases, and then of course we have the backbone in red here, and then we have all of these nucleotide bases attached in this manner. In addition to encoding a functional RNA, a gene also specifies the amino acid sequence in a polypeptide. Now, of course, we're probably aware of the fact that protein is actually made of a combination of polypeptides, and polypeptides are formed from long chains of amino acids. Now, just to drive home the point, we have about 20 naturally occurring amino acid in proteins. Now, it's very important for us to take note of the fact that it's in proteins, because we have more than 20 naturally occurring amino acids. However, in proteins, we have 20 naturally occurring amino acids. There may be other naturally occurring acids, but they do not occur naturally in proteins. Okay, so we have these genes, and these genes basically specify the amino acid sequence of a particular polypeptide that would go on to form a protein. So take, for instance, we have A to say W. Okay, this would be the naturally occurring amino acid in proteins that we have. Now, what a gene does is that a gene would say, okay, the first amino acid in the polypeptide should be C. Okay, C would be the first amino acid in the polypeptide. Now, after attaching C, you attach U. After attaching U, attach T, then attach S, then attach B, attach G, and attach J. It's kind of like a recipe. Okay, it's a protein recipe. It gives the instruction for how they should be attached. Okay, so we have C being attached. This would be the polypeptide that we're cooking up. First is to attach C. After attaching C, you attach U, then you attach T, S, B, then G, and J. Okay, this is how a gene specifies the amino acid sequence of a polypeptide. Okay, so the gene forms part of the DNA. However, the DNA is restricted to the nucleus. In our video on the cell cytoplasm and plasma membrane, we considered the ribosomes, and we saw that the ribosomes serve as site for protein synthesis. Okay, now we have these genes within the DNA, which is within the nucleus. However, the protein or polypeptide amino acid sequence that they specify takes place outside of the nucleus. It means that we would need a messenger to carry this recipe instruction to the site of synthesis. Okay, so we have this recipe, which would be the gene. Now, the gene is just a section of the DNA. So imagine the DNA as this very big cookbook. Okay, so the DNA would be our cookbook. Now, the gene would be a chapter of the cookbook. So we have the gene as a chapter of the cookbook, and it contains the recipe for a particular protein. This gene, which is a chapter of the cookbook, is restricted to the library, okay? And the library would be the nucleus. So we have our library, which is the nucleus. We have our cookbook, and within the cookbook, which is the DNA, we have the gene. Now, the students, so to say, are not allowed to take this cookbook out of the library. It's prohibited. The cookbook is not to be taken out of the library. However, we need the recipe which is contained in the gene in order to produce the proteins. And the protein kitchen is outside the library. So we need a messenger, and that messenger would be the messenger RNA. Now, because the DNA cannot leave the library, which is the nucleus, the messenger RNA performs that function of a messenger as well as a decoder, okay? It performs these two important functions. So just as a recap of all that we have been discussing, we said that the nucleus is kind of like a cookbook library, okay? It's a cookbook library. The DNA would be the cookbook, okay? It contains all the recipes for all the proteins that that the cell would ever need, basically. Okay, and then we have a particular chapter of the cookbook would be the gene, okay, and we define the gene in this very fancy definition as a series of nucleotide base sequence that encode a functional RNA or that specify the amino acid sequence in a polypeptide. Okay, now we said the RNA would serve the messenger function and the decoder function. Now, this function is what we call transcription. So the first step in protein synthesis is the transcription. Now, if we're familiar with some transcription softwares or a transcriber, when you make use of a transcription software, the transcription software converts spoken words into written words. Now, during DNA transcription, what we have is that the information contained within the gene is transcribed, is written down on the RNA, the mRNA. Now, there are three major phases of transcription. First, we have the initiation, then we have the elongation, and lastly, we have the termination. So what exactly is involved in the initiation phase? In the initiation phase, we have what we call a transcription factor. This transcription factor causes for the histone proteins at the transcription site to unwind. Okay, so it causes an unwinding. So if this would be the double helix structure, this is the double helix structure, the transcription factor causes for that transcription site to unwind. Okay, so we have an unwinding that looks something like this. Now, when we have this unwinding, we have the transcription factor binding to what we call a promoter, and this promoter is a special DNA sequence that contains the start point. Okay, it contains the start point of the gene to be transcribed. So it's what tells the RNA. It tells the RNA you can start copying from page 3 of chapter 4. I hope we understand the concept. That is what the promoter does, and the transcription factor binds to the promoter. That is the initiation stage. Okay, that is what we have at the initiation stage. Now, it's important for us to take note of two terms. We have what we call the template strand and the coding strand. Now, the template strand is basically the strand of the DNA molecule that is actually transcribed. So it's the part of the DNA molecule that is transcribed. However, if for instance we have an A here and a T, then a G, and then a C, okay, so we have A, T, G, C. A would normally bind to T, but of course, because we're making use of an RNA molecule, T would be replaced by U. So what we would have here would be uracil, and then T would normally bind to A remains, okay, and then we would have G binds to C, and then C binds to G. So this would be our RNA molecule. Okay, so we have UACG forming the RNA molecule. But take note of the fact that although this is the template strand, we also have the coding strand. The coding strand is the strand that is not transcribed. Okay, now let's take note of the nucleotide bases that are attached to the coding strand. On the template strand, TS, we had attached ATGC, okay? And then forming our RNA, we had our RNA molecule this way, UACG. Now, the coding strand is the strand that is not transcribed, okay? So this would be the coding strand. Now, considering the nucleotide bases on the coding strand, here we would have A attaching to T, T attaching to A, G attaching to C, and C attaching to G. Now, notice what we have in our mRNA. In our mRNA, we have UACG, okay? And in our coding strand, which is not transcribed, we have TACG. Now, notice that the only difference between what we have in our mRNA and what we have in our coding strand is T. And this is because T or thymine is replaced by uracil in an RNA molecule. However, ACG, they are both identical. And so the coding strand is actually somewhat identical to the information that is copied in the mRNA. Okay, so that's why it's called the coding strand, because to some extent, apart from uracil and thymine, it basically contains what we have in the mRNA molecule. However, the template strand is the strand where transcription occurs. Okay, so it's important for us to take note of that. So we have our initiation, where we said we have the transcription factor binding to the promoter, and the promoter is that part that says start copying or start transcribing from page 3 of chapter 4. Okay, so it's that special DNA sequence. And then of course we have elongation, which would be the next step. Elongation being the next step of transcription, where basically we have kind of like a growing mRNA. So the mRNA would continue to grow in this fashion until it gets to a terminal signal. Okay, now the terminal signal is that part that would say, okay, stop at page 25 of chapter 4. That is the terminal signal. So the recipe you need for this particular protein is from page 2 of chapter 4 to page 25 of chapter 4. Do not go beyond page 25. That would be the terminal signal. So the mRNA would continue to copy and copy and copy until it gets to page 25 of chapter 4. Basically, that's how it works. Okay, so we have the terminal signal, and this of course is in the termination phase. So just as a recap, first we have initiation, which is the start, transcription factor, promoter, we have elongation, then we have of course termination. Now it's very important for us to take note of two terms. These terms would be the exon and the intron. Now, what exactly is an exon and what is an intron? Now, an exon is the part of the gene that actually encodes for a protein. Okay, so it would be this series of nucleotide base sequence. Say, for instance, it would be nucleotide base sequence like A, G, C, A, G. And of course would have maybe a C again. Okay, so this would be the exon. Okay, it actually encodes for a protein. However, in the DNA molecule, we have a lot of segments that don't actually encode for a protein. They are physically included, hence the term in, because they are included, but they are not expressed. Now, that part of the DNA that actually contains codes that are included and expressed from the exon, okay, because they are expressed. These are just included, but they are not expressed. Now, just to bring it to view, the exon would be the steps in the recipe, the part that says add 2 alanine, then add 1 methionine. This would be the exon, okay, in the recipe, in the protein recipe. The intron, however, would be this meaningless series of nucleotide sequences, okay, so it could be AG, AGC. AGC is not necessarily meaningless, but this is just an example. Okay, so it would be that series of meaningless nucleotide based sequence, and sometimes they actually interpose. Okay, so we could have meaningful exon. So we have the meaningful exon within the gene, say I would simply use letters. Let's not go into the nucleotide bases. Okay, take for instance, we have CAT. Okay, now interposing between CAT may be something like UIG or UIJ. Now on the other side, could have TARH. This is just an instance. Now notice this interposing section UIJ. Okay, so we have UIJ as an interposing section. So this would be an exon, this would also be an exon, and this would be an intron. UIJ doesn't exactly make sense. Now, of course, we said the gene is transcribed, so the gene is copied. It means the mRNA would actually copy this information. So the mRNA would copy something like CATUIJ Okay, this would be the information copied by the mRNA. Now, the mRNA in this form would be what we call the pre-mRNA. This is the mRNA when it contains introns. Now, when these introns are excised, which simply means when they are cut, okay, so CATUIJTARRH. When the introns are excised, then the exons would be spliced together, so they would be brought together. So would have something like this. This would be the introns physically included but not expressed, and of course we have our exons on the other side. When this part is excised, we would now have our mature mRNA. The process I just explained is what we called mRNA processing. Okay, the process by which the introns are removed and then the exons are spliced together. So now we would have a meaningful word, for example, and the word once UIJ is removed would be, okay, so we would have CATTARH. Kata is not an actual word, but this is just to bring it to perspective so we can understand. But say for instance it's an actual word, and then this would be mRNA processing. So we excise the introns and then splice the exons, okay, mRNA processing. So once the mRNA has been processed, it's exported. Okay, so it's exported out of the nucleus. Okay, it's exported out of the nucleus. So now we have our mature mRNA. Now, so far we have been considering mRNA, which is messenger RNA. There are two other forms of RNA. This would be the tRNA, which is the transfer RNA, and of course we also have the ribosomal RNA. And we said the ribosomal RNA complexed with proteins to form ribonucleoproteins is what forms the subunit of the ribosomes. And we said it is synthesized ribosomal RNA. Unlike mRNA and to some extent tRNA, ribosomal RNA is synthesized in the nucleolus. Not the nucleus, but the nucleolus. Okay, and then it forms part of the ribosomes. Now, as we'd go on to see, the tRNA is kind of like L-shaped. It's an L-shaped molecule. On one side, it has anticodons, and on the other side, it has the amino acid. We'll go on to see this in details as we go on. So on one side, we have the amino acid. On the other side, we have the anticodons. Now, we have our recipe transcribed. Okay, our recipe has been transcribed into the mRNA, and now it's going to the protein kitchen, which would be the ribosomes. Okay, now what we have we have the ribosome being made of two subunits. So we have the large subunit and we have the small subunit. So this is the small and this is the large. Okay, now earlier on I stated that the transfer RNA is this L-shaped molecule and it contains the anticodon on one side and then the amino acid for that for the codon on the other side. What controls the attachment of the amino acid to the tRNA is an enzyme called the amino acyl tRNA synthase. Okay, it's an enzyme that basically controls the attachment of the amino acid to the tRNA. When the amino acid is attached to tRNA, what is formed is an amino acyl tRNA. Okay, so it's important for us to take note of these terms. So we have our information that has been transcribed. Okay, the recipe has been transcribed. Now what we need is translation. So the translation is the next step. Translation. Just like transcription, we also have 3 steps taking place in translation. We have initiation, then we have elongation, we have termination. Now, as I stated earlier, the ribosome, we have the large subunit and the small subunit. Now, within the large subunit, we have 3 sites of attachment for the tRNA, 3 sites of attachment for the tRNA, and 1 site of attachment for the mRNA within the groove of the large and small subunit. So the mRNA would be within this groove. So this would be the mRNA in white. Now the 3 sites would be the A site, the P site, and the E site. Now A site is simply the amino acyl site. The P site is the peptidyl site, and then the E site is the exit site. Okay, so very quickly let's take a look at this. So this would be RNA, the large subunit, E, this would be E, this would be P, and then of course this would be the A site. Now what happens is that the ribosomes would scan through the mRNA. Now the mRNA is made of codon. Now the codon is simply a series of 3 nucleotide bases. A series of 3 nucleotide bases that code for a particular amino acid. So an example would be AAA. This would be an example of a codon. It's a triplet. Okay, so basically three nucleotide base form the codon. Now earlier when we considered the transfer RNA, I stated that we have an anticodon on the transfer RNA. Now a transfer RNA anticodon for AAA would be UUU. Okay, so it would simply contain the complementary base pair for the code of the mRNA. Okay, now we have what we call the start signal. Okay, remember when we were considering transcription, the start was the promoter. Now the start signal for the initiation in translation is a particular codon, and it's the codon that codes for methionine. Let's take a brief look at this codon dictionary or protein dictionary, so to say. Now here in this table we can see that we have the start codon as AUG. Okay, AUG. How I remember it is I simply remember the word augment. So augment means to kind of like increase by adding, increase an effect or increase a particular thing by adding to it. This is not a standard definition, but augment has to do with increase, and the start would eventually lead to an increase. Okay, so basically it's how I remember it. If it helps, you can also adopt that method of remembering. So augment, AUG, and it codes for methionine. you may say, well, since methionine is the starting codon, does this mean that every protein has the first amino acid as methionine? The answer to that question would be no. And this is because although methionine indeed forms the very first amino acid for the polypeptide, after translation, the methionine is usually cut off. Okay, it's usually cut off. And it's also important for us to take note of the fact that the codon AUG is not necessarily the first codon in an mRNA sequence. So we could start with, say for instance, AAA. Now what happens when the mRNA starts with AAA is that the ribosome would basically scan through the mRNA. So the ribosome would scan through the mRNA until it gets to the point where it reads AUG. Okay, when it reads AUG, then the transfer RNA, which holds methionine, would have the anticodon UAC. Okay, it would have the anticodon UAC, and it would sit at the amino acyl site. So it would sit at the amino acyl site and merge with AUG on this spot. Okay, so we would have methionine. Now what happens is, after methionine has been added, the ribosome would translocate. Okay, so it kind of like scans for the next codon. When it scans for the next codon, and say for instance the next codon is an AAA, okay, going by our table here, AAA codes for lysine. Okay, so AAA codes for lysine. Now of course we already have our methionine. We have our methionine on its transfer RNA. This would be the E site, this would be the P, and this would be the A site. The methionine with the transfer RNA is now moved to the P site, because as the ribosome translocates, the transfer RNA also moves progressively from A to P and then to E. So we have our methionine here. Okay, and then we have, or the next codon now would be the AAA, and we said it codes for lysine. Okay, so what we would have this tRNA molecule that would have the anticodon UUU. So UUU would merge with AAA on the mRNA. So we'd have the tRNA here. So now we're done with initiation, and initiation starts with the initiating codon, which is AUG. Okay, now we're going on to elongation, which is the next step of translation. Okay, so we have lysine. Okay, we have lysine there. Now what happens, this is of course the A site. Now what happens is that methionine, or the growing chain, let's assume we already have a growing chain. The growing chain of amino acid is transferred. The growing chain of amino acid would be transferred to the A site. So we'd have something like this. And then this transfer RNA at the P site would be moved to the E site. Then would have this transfer RNA moved to the P site, and then the chain would continue to grow in that order. Okay, so first we have initiation by the AUG codon. Now let's take a brief look at termination. Now we have what we call stop codons. These stop codons do not code for any protein. They simply indicate when the ribosome should stop synthesizing. Basically we have three stop codons. We have the UAA, the UAG, and then the UGA. Now how I remember it is this: U is always first, so U would always be first. The next rule I used to remember these stop codons is that G cannot appear twice. So G cannot appear twice, and A can appear twice. So to remember the stop codon, U would always be first, so we'd have U. G cannot appear twice, so if we have a G, we'd have to have an A. A can appear twice, so we'd have a U, AA, and we could have a UAG. Okay, these are the stop codons. That's the memory tip that I used to remember these stop codons. The sub steps of elongation would be the codon recognition, then we have a formation of the peptide bond, and then we have the translocation of the ribosome, where the ribosome scans again for the next codon and so on. The ribosome gets to, say for instance, a UAA, which happens to be a stop codon. This stop codon doesn't code for any amino acid. So what happens is that it fills in this A site with water. Now remember the term hydrolysis, which has to do with using water to break down bonds. Okay, now earlier I stated that we have a bond between the transfer RNA and the amino acid that it holds. Okay, and that bond is the amino acyl transfer RNA tRNA bond. Now basically, when the water is added at the A site due to the stop codon, that water molecule helps to break down the bond between the tRNA and the forming peptide chain. Okay, so basically the two subunits separate, and then what we now have is that we have a peptide chain. Okay, so we have a peptide chain and the two subunits separate. Now it's also important for us to take note of the ER signal sequence. Now not every forming peptide chain has an ER signal sequence. Okay, some forming peptide chains have the ER signal sequence, and these peptide chains are mainly the peptide chains that form secretory proteins. Now ER stands for endoplasmic reticulum signal sequence. It's simply a sequence of amino acid. It's a short peptide, okay, and it basically signals that this particular protein is to be taken to the rough endoplasmic reticulum. So when the protein begins to form at the ribosomes, if it happens to have an ER sequence, then the ribosome gets attached to the endoplasmic reticulum by what we call the SRP. SRP stands for signal recognition particle. Okay, so it's important for us to take note of some of these terms. And of course, when the protein is meant to be taken to the endoplasmic reticulum, mainly secretory proteins that remain in the cytosol are synthesized by free ribosome. Okay, so it's important for us to take note of that. Now, very quickly, having considered protein synthesis, which we said takes place in two steps, transcription and translation, let's take a look at our outline. Alright, so going by our outline, we have considered protein synthesis. Let's take a brief look at cell renewal. Now, for cell renewal, we have what we call the non-renewable population. We have the continually or continuously renewing population of cells, and then we have the potentially renewable population of cells. This simply has to do with how cells are able to renew themselves when they become worn out. So it's important for us to take note of the fact that as cells specialize, they actually lose their ability to reproduce. They lose their ability to divide and form new cells. They lose their renewal ability. So the more specialized a cell is, the less likely it is for the cell to proliferate or to divide. So this sort of cells would form the non-renewable population of cells. They are highly specialized cells. The continually renewing population of cells are those cells that are either progenitor cells. So they are progenitor cells. When they are progenitor cells, they are less differentiated. Okay, of course, differentiation and specialization work hand in hand. So progenitor cells are usually less differentiated, and because they are less differentiated and by implication less specialized, it means that they still have the potential to divide and proliferate and renew. Okay, so we could have progenitor cells, and we could also have stem cells. Stem cells are continually renewing cells. They are not differentiated. For the stem cells, they could be pluripotential when they are able to form more than one type of cell. So we could have pluripotential stem cells. We could also have unipotential stem cells. Unipotential stem cells are only able to proliferate into a specific type of cell. It's also very important for us to take note of the fact that stem cells are actually continually renewing, and they do this by a conservative form of division. So we have what we call the self-renewal division of stem cells. Now, the cells produced by the self-renewal division are not differentiated. So these cells remain stem cells. So if we have a stem cell like this stem cell divides. So this, for example, A could be the self-renewal division cell. This cell remains as a stem stem cell, and then we have the differential division, okay, or we call it the differentiative division. The cell formed by differentiative division are the cells that would go on to form differentiated and specialized cells when the cells become worn out. And of course, we have the potentially renewable population of cells. The potentially renewable population of cells are specialized cells that can regain their renewal property when the need arises. For example, we have the liver cells. Now, in a case where we have a mass of the liver that is cut out, the liver cells are specialized cells, and on a normal condition they would be non-renewable cells. However, when a mass of the liver is cut out, these cells actually have the potential of regaining their renewability, and so they are able to proliferate and kind of like regain the mass of the liver that has been cut out. So that's just a brief overview of what the cell renewal is all about. Now, for the remaining parts of what we have to consider, the telomere or the telomerase, the telomere and telomerase. Now, the telomere is actually a special sequence of DNA nucleotides. So we have our chromosome that looks something like this, okay? Of course, we have the centromere. Now, the telomere is that part, it's a special nucleotide sequence at the end of the chromosome, and it prevents the chromosomes from fraying, okay? Now, as a cell divides, proliferates on and on, what happens is that the amount of telomere present decreases over time, and so a cell is only able to divide for about 50 to 90 times because the telomere continues to decrease, okay, as cells divide. Now, telomerase have the ability to renew the telomere, and this is a property of cancerous cells. Cancerous cells actually have this telomerase, and the telomerase is able to revert the process, okay? So we have a kind of like a renewal of the telomeres, and so the cell is able to divide infinitely, okay? So that's that about the telomeres. Then we have the idea of apoptosis. Apoptosis is a programmed cell death, okay? Apoptosis is a programmed cell death, and on our video on mitochondria, we stated that the mitochondria does play a role in apoptosis. So what happens is that in apoptosis, we have that the mitochondria becomes very permeable. When the mitochondria becomes very permeable, it releases the enzyme cytochrome C. Now, cytochrome C, when it's released into the cytosol, it activates what we call the caspase, and these caspases would cause for digestive activities to take place within the cell, okay? And then when the digestive activities take place within the cell, what happens is that we have a bulb, okay? A bulb of these digested cells would be formed. These digested cells would then send the signal to macrophages, and these macrophages would then ingest and phagocyze the destroyed cell, okay? So that's the general idea of apoptosis. Now, our next subtopic is karyotype. Karyotype simply has to do with the entire chromosomal makeup of an organism or of an individual. It's simply the complete set of chromosomes that we have in an organism, okay? Now, very quickly, let's take a look at some clinical correlates of all that we have learned so far. The first clinical correlate would be considering is aneuploidy. So generally, taking humans, for example, we have 46 chromosomes, okay? This is said to be diploid. We have 46 chromosomes, 23 pairs of chromosomes. Alright, so when we have 23 chromosomes, for instance in sex cells or gametes, when we have 23 chromosomes in gametes, this is said to be haploid, okay? Haploid. Now we have diploid, which is 2n, and we have haploid, which is n, okay? We could also have polyploid, okay? We could have the term polyploid, where we could have xn, where x could be very large numbers like 10, like 20, and so on in a particular cell, okay? For example, in megakaryocytes, we have about 23 times the number of chromosomes that we have in a diploid, okay? So this would be about 64n, okay? This is polyploidy. So generally, we have a multiple of the haploid. So it could be n, or 1n, 2n, 3n, and so on. Now, what happens when we have a number of chromosomes in a cell that is not equal to a multiple of the haploid? So the haploid is 23, and then we have 46, and so on. What happens when we have 45 chromosome number? Or say, for instance, we have 47. These two numbers are not multiples of 23. When a situation like this occurs, what we have is aneuploidy, and specifically in humans it could be in the form of monosomy or trisomy. Monosomy would usually be associated with number 45, and trisomy would usually be associated with number 47. An example of trisomy would be trisomy 21, which is associated with Down syndrome. Now, generally we have in females 23 pair of chromosomes, 23 pair of homologous chromosomes. So we have 22 autosomes, and then we have one pair of allosomes or sex chromosomes. Same in male, we also have 22 pair of autosomes, and then the allosomes for the male of course would be XY. For the female we have the XX. Now, this is kind of like a rule, and the rule is that when we have more than one X chromosome, okay, so when more than one X chromosome is present in a particular cell, like in females we have the XX, one of the X chromosome would be condensed and the other would be extended, meaning it would be transcribable. So in other words, one of the X would be expressed and the other would kind of remain dormant. Okay, this is when we have two X's, like in females, XX. This brings us to certain other conditions such as the Klinefelter syndrome, which occurs as a result of non-disjunction. Now, in Klinefelter syndrome, due to non-disjunction, we have the XXY. Okay, notice that we have two X chromosomes, and as the rule goes, when we have more than one X chromosome present, one would be transcribable and the other would be condensed. This means that a male that is born with an XXY would exhibit some female characteristics because of the other X that is expressed. Generally, when a normal male is born XY, we don't have two X chromosomes. So since there are no two X chromosomes, the X would not be expressed at all. However, we have two X chromosomes present here, and so one of the X's would be expressed alongside the normal male characteristics. So this would be Klinefelter syndrome. We also have the Turner's syndrome, which is an XO. Okay, so what happens here is that this of course is in females, so the other X that would have remained condensed is absent. That's why we have an O here instead of an X. Okay, and so the female is born sterile due to the absence of that X. Alright, so far we have considered the nucleus. We said the nucleus is the information center of the cell, kind of like the CPU. We considered the nuclear envelope. Then we went on to see the nucleoli. We went on to see the nucleoplasm. We went on to see the cell cycle, which we said is divided into the interphase and the cell division. We said the interphase is divided into the gap 1, the synthesis, and the gap 2. And we said cell division is in two stages: mitosis and cytokinesis. Okay, we went on to take a look at DNA replication, under which we saw the uncoiling, then we saw the separation, the assembly, and the restoration. Also, we went on to take a look at mitosis under the heading of prophase, metaphase, anaphase, and telophase. Then we considered protein synthesis, where we considered translation and transcription, both of which have initiation, elongation, and termination as sub-steps. We went on to see cell renewal. We considered non-renewable cell population, potentially renewable cell population, and continually or continuously renewable cell population. We went on to see the telomeres, which we said are a special sequence of DNA that occur at the free ends of the chromosome. chromosome and determine how much a cell is able to divide. We said telomerase is an enzyme that is able to kind of renew the telomere. Went on to see apoptosis. We said apoptosis is a programmed cell death and the mitochondria does play a role in apoptosis. Also, we saw that the karyotype is the chromosomal makeup of an organism. We went on to see clinical correlates like aneuploidy, where we consider trisomy and monosomy, and then we went on to see the Klinefelter syndrome, the Turner syndrome, and also Down syndrome. This has been a very long video. If you liked this video, please give it a thumbs up and don't forget to subscribe. Thank you for watching till the end. Bye.